on resources. So as of right now in Illinois, we have a total of 68 counties that have been confirmed to have EAB, and this is likely an underestimation. There are probably more counties that we could light up the map with, but we just don't have the confirmed specimen just yet, but that's something that we're going to be looking to really focus on over the next few years. And to this point, the major management strategies have been things like treating trees. So for instance, the city of Chicago treated their ash trees. There's also tree removal, whether that's to try and get ahead of potential infestation by removing host material, which is really, we're way past that now. More often than not now, tree removal is in response to a tree being dead and dangerous, a liability as it might drop limbs, injure somebody. There's also regulations. So anybody who's bought firewood or gone camping has probably heard about how you should not be moving firewood around where you didn't harvest it from, things of that nature. We're all really familiar with the situation at this point. But moving sort of into these next steps, the agency, a big focus is biological control. And biological control has been around for quite a long time, over 100 years. Unfortunately, it did get kind of a negative reputation for itself because a number of projects were undertaken that had unintended consequences, right? Anytime you're introducing a non-native organism, there's the potential for problems. But our project skirts around many of those concerns. It goes above and beyond to make sure that this is a safe program, that this is an effective program, and I'm going to talk a bit about how we make all of that work. So generally speaking, biological control has certain steps that you have to follow to make sure that it's done right, that it's done properly. And first and foremost is you have to learn about the pests that you're dealing with, right? So when EAB first arrived on the scene, lots of research was done pretty much immediately to try and figure out elements of its life history, host specificity, weaknesses, things that we could use to potentially treat the problem. And then the other thing that happens is when it comes to a new place, you look around to see, well, are there any native taxa, organisms, et cetera, that could help us deal with something like EAB? So there are a number of native parasitoid wasps, those 4 species on the right side of the screen. And these traditionally feed on native buprestid species. So the jewel beetles that are already naturally occurring here in North America and the US more specifically. And unfortunately, the research that they did demonstrated that these 4 really weren't able or interested to do the kind of work that we wanted them to do, right? They simply just did not really respond to EAB in the way that we would want a parasitoid to respond. So the next step then is to go to the source. EAB comes from Asia, and so the idea is you go over there and you look for the things that prey on EAB in its native habitat. And from that really broad umbrella, you look for the most likely candidates that you think would work best as biocontrol agents. You bring them over here to the US under really strict import conditions. You study them in quarantine. You're making very sure that nothing is getting out before you're ready for it to be out in the world. There's a lot of research, a lot of work that has to be done to draft up what we call release justification. So these would be things like environmental assessments demonstrating that this parasitoid wasp is highly specific, is unlikely to go after anything that is not EAB, all above board. There's a lot of permitting that has to be done because, again, anytime that you're releasing something non-native, you want to be very careful and make sure that you have the appropriate permissions and permits. Field sites need to be selected. There needs to be a mass rearing program in order to provide all of the folks who want these wasps, these parasitoids, with the appropriate supply. And And then on the tail end of things, you want to take a look at whether or not, one, the releases are working. Are these parasitoids becoming established? And then two, if they're becoming established, are they having the kind of impacts that we want to be seeing from them? So the EAB Biocontrol project has been going on for quite a while. The first releases began back in 2007. And this focuses on 4 natural enemies of EAB, all small parasitoid wasps that have been approved for release. They were found to be significantly specific to EAB. They don't go after anything else. And they are raised in a rearing facility out in Brighton, Michigan. To date, they have reared and released over 5 million parasitoid wasps, and that's across 27 US states, as well as 2 Canadian provinces. Now, the goal of a program like this is clearly not going to be eradication. We're very far past that point, but the hope is that these parasitoids can help to maintain or control EAB populations at a more manageable level, and perhaps leave some breathing room for ash regeneration to occur in some of our native areas. When releasing parasitoids, site assessment and selection is very, very important. These wasps need every possible chance they can get to. They need a lot of support, right? We want to give them the best possible chance of becoming established. So the research that's been done has demonstrated that we like sites that are at least 40 acres, if not larger, and contain a pretty high proportion of ash. 25% is sort of the basement that they like to see, but more obviously would be better. And there is wiggle room here if a site is smaller than 40 acres, if it happens to be connected to other applicable areas by perhaps a waterway, a forested corridor, anything that would allow easy dispersal of the wasps. Sometimes there can be exceptions made for the acreage requirement. We require that the site not be disturbed, harvested, treated, etc. for 5 years. And that's to ensure that the wasps are not inadvertently killed at the site. We want to make sure that any data we're collecting from these efforts accurately reflects what is actually going on and isn't confounded by tree removal, tree treatment, etc. It's important that there's a range of ash size classes. The parasitoids all have different morphological characteristics that make them better suited to, say, smaller branches with thinner bark, or perhaps the thicker bark that's on the trunk of an ash tree. And furthermore, the large ash trees tend to be really great reservoirs for lots of larvae, lots of eggs, and that helps to get the wasps established. But the small understory ash, the seedlings that are just coming up, are important for supporting the parasitoids after the EAB crash, right? So when all of those large trees have kicked the bucket, those small seedlings are going to be the next reservoir for EAB, and therefore the next reservoir for our parasitoid species. Last but not least, we want, obviously, we want EAB to be at the site in order to support the parasitoid's growth and development, but you don't want there to be too much. We want sort of a low to moderate level, and this can be tricky. Oftentimes, EAB infestation, we don't catch it until it's pretty well developed. The early symptomology can be tough to miss. But ultimately, we want a site that's mostly 2s and 3s in regard to canopy cover, or I'm sorry, crown class. So when we have a site in mind, the first thing that we do is we submit it to a website called Biocontrol. This is nationwide. This supports all releases all over the country. And we submit our sites through Biocontrol. They take a look at the site's characteristics, and they determine whether or not they think it's a good fit for the project. The next thing that we do is when we're actually performing releases and then recovery a few years after that, all those data get put into Biocontrol so that they can keep tabs on the progress of the project and look at the outcomes. And another great thing that Biocontrol is able to offer is it takes all of those data that have been put in from all over the country and it maps them. So that map there at the bottom is every single parasitoid release tree as of just this month. So every single one of those green dots represents an ash tree that had parasitoids released on it in some way, shape, or form. So I've talked them up quite a bit. Let's actually meet our parasitoids. Our first one is a teeny tiny little parasitoid called Oobius agrili. This is an egg parasitoid. So an adult female wasp will lay her egg on an EAB egg. You can see that just here. This is the EAB egg, the brown area. And you can see she's inserting her ovipositor into the egg to lay one of her own eggs inside of it. And when that egg hatches, it'll parasitize the EAB egg, kill it, and yields a new adult wasp that will start the process all over again. And Oobius is parthenogenic, so this female is only going to have daughters who will then go on to have more daughters. Research has demonstrated that Oobius does a pretty good job. It parasitized up to 60% of the EAB eggs in a study that was done in China. And one single female was found to be able to parasitize up to about 80 EAB eggs during her lifetime. The problem is, though, that it's a little bit limited in that one egg is only going to generate one wasp. You'll see our other parasitoids have a bit of an advantage when it comes to reproductive rate. The other disadvantage that Oobius has is that it is very small and it is the weakest flyer of the four. So its dispersal is a little bit more limited than the other species. That we can release all over the country. You'll see in just a few slides that some of our other parasitoids don't do well in cold temperatures or warm temperatures. They have specific life history needs that have to be met. And Oobius, we release it at about 600 growing degree days. So we want to target the appropriate life cycle stage. And that's about when we have the highest probability of there being lots of viable EAB eggs for these wasps to parasitize. So when we release these out in the field, they come in these repurposed medication bottles called Oobinators. I think that's probably a proprietary term that I cannot take credit for. But you'll see there's some sort of mesh on the bottom and there's filter paper folded up on the inside that has parasitized EAB eggs stuck to it. So when the Oobius hatch, they come out of the eggs, they exit the Oobinator and then move out into the release site. And as I mentioned, the scale on these things, these are very, very small, half a millimeter to a millimeter at most. Our next candidate is Tetrastichus planipennisi. This is sort of the star of the show. Tetrastichus traditionally does very well. This is a larval parasitoid, so rather than going after eggs, this wasp is going to go after EAB larvae that are in the host tree. They're internal feeders, so a female Tetrastichus will use her ovipositor to punch through the bark of the tree and lay eggs in EAB larvae. Those larvae will then feed on the EAB larvae from the inside out. And one larva can support up to 130 adult wasps. So you see this just has a definite advantage when it comes to making more of itself over Oobius. The only problem is that the ovipositor on this species is on the short side, so it does best when there are host trees that have 6 inches or less. At DBH, we need the bark to be a little bit thinner so it can actually reach those EAB larvae. And this one, we release it in counties that accumulate around 3,500 growing degree days or less. Anything more than that, and it decreases the chances that there's going to be the appropriately sized larvae for it when it comes out in the spring. We do a couple of releases on this one. We start sort of early season. That's when it's going to be attacking the overwintering EAB larvae. And then later in the growing season is when it's going to sort of try to catch up with the new generation of EAB. And Tetrastichus tends to be released from bolts. So we get sections of ash that are full of parasitized EAB larvae, and the wasps emerge from the bolts. Our final two parasitoids are both in genus Spathius. Spathius agrili, this is one that I do not have direct experience with because of my location here in the state. I'm too far north. But this is another larval parasitoid, except this one feeds externally. So similarly to Tetrastichus, the female is going to lay eggs on an EAB larva, and those larvae are then going to remain on the outside of the EAB larva and consume it from outside in. So different from Tetrastichus. You can see in the photo here, it has a longer ovipositor. So these are the ones that are able to fill those gaps left by our other parasitoids because they can get through thicker bark. This species, we only release it south of the 40th parallel, and that's again due to growing degree day accumulation. It's just not something that does very well this far north in the state. Further south, it seems to do okay, but when we want to release Spathius, at least in my neck of the woods, we release Spathius gallinae. This is another larval parasitoid, very similar to the preceding species. Major difference is that it is much hardier. It can handle colder temperatures. This is from area of Russia, I believe. So it's much more well suited to the temperatures that we have up here in this area. And Spathius tends to be sent to us as adults. So we will get plastic cups full of adult wasps, and we have to treat them very carefully, handle them very gently, and we take them out to the release site and put them onto host trees to give them a chance to find some EAB to parasitize. So when it comes to doing a release, once we receive the wasps, there's guidelines that should be followed to give them the best possible chance of establishment. You want to pick the right kind of ash trees. You obviously want there to be some level of infestation. There needs to be a food resource for the wasps to consume. You also want to disperse the parasitoids throughout the site pretty widely so that they're not having to do all of the flying on their own. It's much easier for you to help them out that way. The timing is important. As I mentioned, we release different wasps at different times because of the developmental stage of the EAB that are at the site. You don't want to release Oobius, an egg parasitoid, when there's predominantly larval parasitoids around. It's just not going to work well for anybody. The guideline states that we want to release at least 200 female wasps every 2 weeks for a period of 6 weeks. That's minimum, but depending on the supply that our rearing facility in Brighton is able to provide, we're often able to go above and beyond that. And clearly, the more you release, the better chance you have at establishment. And there are a number of other considerations that need to be thought about as well. It's better to, for instance, release wasps in the morning or evening when they have a chance to be shielded from hot midday temperatures. It's not a great idea to release them when it's really windy. It's not a great idea to release them when it's rainy. You have to be careful when handling them if you have bug spray on your hands, if you're wearing permethrin treated clothing while you're out in the field. All of these things, these are pretty delicate organisms, and the estimate is that it costs between $4 and $6 to rear and ship and release every single one of these wasps. So they're rather expensive for tiny little things, and we try to be as careful with them as we can. So here in Illinois, releases have been happening. The first ones were back in 2009, and those were releases that were performed not by us. So this would have been, Cook County, Lake County, DuPage County. Green Valley Forest Preserve is a great example. Fredric Miller can talk quite a bit about that. He was very involved with those releases. Those releases happened between 2009 and 2016. And then our involvement in this project began in 2018. So you can see we have a number of sites where we have performed releases, 22 counties. And the map on the right gives you a specific idea of where those are, as well as how many wasps were released. So we have a number of sites that have gotten quite a number of parasitoids released. That spot in the very middle of the state, for instance, that's down in Macon County, over 25,000 wasps just at that site alone. In total, we released over 213,000 wasps, and the vast majority of those are Tetrastichus. This has a lot to do with the ease of shipping Tetrastichus. It's really well protected in a bolt. It has to do with the ease of rearing Tetrastichus. As I mentioned previously, you can get quite a few wasps out of one EAB larva. So you're just going to have a much easier time making them and shipping them, and that's why we release so many of them. So about 62% of all of the wasps that have been released in Illinois from 2009 to 2020 are Tetrastichus. So once the releases have been performed for 2 years, the third year is devoted to recovery. We want to take a look at whether or not things have become established. And there are a number of methods that can be used to determine this. One of the first that folks can use is to peel ash trees looking for the larval parasitoids. So this is just manual labor, right? You cut the tree down, you cut it into bolts, and you peel the bark looking for EAB galleries, looking for evidence of larval feeding, right? So you may see Tetrastichus feeding inside an EAB larva. You can see them. This larva is just full of Tetrastichus larvae. You may see evidence of Spathius. These are external feeders. So there is a tiny little remnant of an EAB larva right there, but it's been consumed by these Spathius larvae. You may see pupae. So Spathius spins silken cocoons and pupates before it turns into an adult. And Tetrastichus also pupates, but they do it asynchronously. So you may see pupae and larvae at the same time. You might also not find the parasitoids themselves, but you'll find evidence that they were there. So meconium is waste. You may see these dark spots in the EAB gallery, and that was left behind by Tetrastichus larvae. Problem being, this peeling process is pretty labor intensive. It takes quite a long time. You need somebody to be able to cut down trees for you. It's a great way to look for them, but it can take a lot of effort, a lot of work. Debarking is similar. This is one of the good ways to look for Oobius, or egg parasitoid, simply by taking a draw knife and scraping off that top layer of bark off of an infested ash tree, and then sifting through that, looking for very small EAB eggs, and in particular looking for ones that are dark in color, like this one here in the center of the screen in the red circle, as well as the one over here. And the red arrow is pointing to a tiny little exit hole that's been chewed in that egg. So there was an Oobius adult in there that chewed its way out. And this photo down here is just pretty neat. You can see very clearly the developing wasp inside of that EAB egg. This is also pretty labor intensive, and because Oobius is so small and EAB eggs are so small, it's very easy to miss them this way, but it's definitely a viable way to do it. There's also the option to place the scraped bark material into a rearing chamber and to see what comes out of that material. The method that we use thus far is yellow pan trapping. This is really effective. Most hymenopterans, wasps, bees, ants tend to be attracted to the color yellow. So this is literally just yellow pan trapping is just 2 plastic yellow bowls, a shelf bracket attached to a host tree, and the bowl is filled with propylene glycol, which is kind of viscous and sticky. And so when wasps are attracted to it, they land on it, they can't leave. We go out and we check those traps once a week during the field season, and then we dig through those samples for evidence of parasitoids. So the photos that you see here, you get a whole mess of hymenopterans, and you have to try and pick through and find the ones that you're looking for. So this one here is Tetrastichus. This is likely a Spathius, and this one down here is Oobius, tentatively. Our samples for this year are still being confirmed, but I do consider that to be a pretty strong suspect. This is a really inexpensive process. It's really effective. It does not take nearly the effort, which is great for us here in Illinois. We're pretty short-staffed, and as we all know, COVID has complicated just about every element of everything. So yellow pan trapping was a really great way for us to address this issue of whether or not we've got establishment at a couple of our sites. So we were able to do recovery at our 2 sites this year. One was that big site down in Macon County, and those samples are still being screened, so I can't report on them just yet. But I can tell you about the recovery that we did in Boone County. We set 15 yellow pan traps throughout the site. The map on the right side, each one of those orange spots or red X's is a trap. And then each of the green dots is a release tree. So that was a tree that at some point in the preceding 2 years had some kind of release performed on it. And we had pretty good success. Granted, these are all preliminary IDs, so it could change a little bit, but it would appear that the majority of our traps caught at least one parasitoid wasp, the majority of which were Tetrastichus, which again, we would expect given their reproduction rates, given the vast number of Tetrastichus that we released at the site. And it tracked pretty closely with when we see the most EAB as well. So we caught the most parasitoids in August. All told, we picked up 37 parasitoids, and again, I mentioned 92% of them were Tetrastichus. One Oobius that I can think of, and a couple of Spathius, but otherwise it was all Tetrastichus all the time. So according to those results, then we can feel pretty confident that Tetrastichus is doing pretty well, is present throughout the Boone County site. Research indicates that it should continue to do pretty well over time, and we're hoping that it'll continue to increase and have the impacts that we want. To date, you know, we're cautiously optimistic, right? So Oobius and Spathius, we did, I think, catch them at this site. There's a possibility that we've caught them at our Macon County site as well, but it's probably a bit early to make a definitive call on these 2 species. And, you know, again, this has to do with their lower reproductive rate. They don't disperse as well, and we just don't release as many of them. So they may take a little bit longer to get a foothold. So looking ahead, here in Illinois, it's going to depend a lot on what our rearing facility is able to provide, as well as our personnel situation here in the office. We have a number of sites that have had 1 year of releases that are going to need a 2nd year before we can perform recovery. Those are all of the yellow counties on the map. And then we've got an additional 10 counties that are due for recovery survey in 2021. So to my mind, that's going to be our biggest focus, is figuring out whether or not those 10 counties have the same positive evidence of establishment that we saw in Boone County. Additionally, you know, we're looking to fill in the map, right? So we're always on the lookout for new sites. Brighton, the rearing facility, emphasizes new counties over old ones. So if we have an existing release site in a county, it's unlikely that they'll want to push through another one. They're more interested in filling the state as best they can and getting really good coverage. Unfortunately, there's actually a lot of research that's been done on these releases because they've been going on for so long, and it has a lot of promising things to say about the potential outcomes we might be able to have here in Illinois. So there has been evidence that Tetrastichus, again, traditionally does really well and does a good job dispersing to areas with high population densities of EAB. So here on the map, these 3 yellow triangles indicate where they released Tetrastichus, and then these, you know, red areas, these hot spots, that's where the highest concentrations of EAB were found. They rate that as severely infested. And the red stars show you where they were catching the parasitoids during their recovery efforts. So you can see, even though they were released, you know, only at 3 spots, they got pretty good coverage all along this corridor here. So in short, they go where the EAB are, all on their own. They do a pretty good job on that. They're also able to move pretty far under their own power. About 5 years after release, they were found over 10 kilometers away from their initial release site. And then in addition to doing a good job dispersing, they are doing the work of parasitizing like we expect them to. So the percentage of parasitism of EAB by Tetrastichus 4 years post-release hit about 11.5, almost 12%. And then the number of trees with at least one brood of Tetrastichus got all the way up to 41.7% 4 years post-release. So that's all very promising stuff. They're getting established. They're having new broods, reproducing, and hopefully continuing on that trajectory. When they get established, it's looking really good that they are actually killing late instar EAB larvae. So this study found that they were able to induce mortality in anywhere between 36 to 85% of late instar EAB larvae. And in this situation, they were looking at the population growth rates. So that's the R sub 0. And 1 is just maintenance, right? So not declining, not growing, just maintaining at a pretty consistent level. And what they did was they did a bunch of modeling, and they found that when you removed Tetrastichus parasitism, those values doubled. So that would indicate that parasitism was able to reduce the pest population of EAB at this particular site by about 50%. We've also got this is not my favorite graph, but I'm going to break it down for you. So we want to focus on the solid lines. This top one here, this is what they call the baseline situation. Again, this was where they modeled a number of different scenarios in regards to EAB infestation and parasitoid releases. So this is baseline. This is you know, a single introduction of EAB. It gets established, it causes problems, and that's it. This bottom situation is what they call the catastrophe, which refers to numerous repeated reintroductions of EAB. Just the site just keeps getting hammered over and over and over again with new EAB infestation. The situation in the middle, this is what they call the reduced catastrophe scenario. So this simulates the effects of biocontrol, so Tetrastichus being released at a site, combined with predation by woodpeckers, right? So they're going after the grubs underneath the bark of these ash trees. So you see it kind of falls out right in the middle. So what this is saying is that it's not obviously, well, let me rephrase that. So when we have the catastrophe and the baseline situation, you can see that they ultimately end up to be flat, right? So there's not growth in the average abundance of ash. So this is looking at ash abundance. This one in the center, though, is the only one that is on a slight upward trajectory. So taking a look at that, the probability of decline, ash decline, was reduced by about 52% in this model when they introduced the impacts of parasitoids combined with woodpecker predation. And then big picture, this is promising in that if we are able to give ash regeneration some breathing room to make that happen, we can help protect understory. So when ash is extirpated from an area, it opens up the canopy. Lots of invasive species are able to exploit those openings. But with the biocontrol scenario, we're going to have ash saplings outcompeting the invasives. They're going to increase shade. They're going to allow shade tolerant natives to continue filling in the area and preventing a lot of invasive understory vegetation from taking over. So it's all looking pretty positive literature wise. We're very excited to continue monitoring the situation here in Illinois. And with that, I would love to answer any questions that folks may have, and I appreciate your time and your attention.
Thank you, Nick. We do have a few questions that have come in, and we'll take a few minutes to do those.
I'll do my best.
Okay, great. The first one is, and you talked a little bit about this, that there's an interest to expand these plantings of these biocontrols across the state. If a landowner is interested in getting involved, how would they go about doing that?
The best way I would say is you can get in touch directly with me. I'm sort of the interim point of contact for this program. It predominantly is going to be run really strongly out of the southern and central part of the state. Frankly, Northern Illinois, we're a little bit past the point where we want to be doing too much release work. Southern Illinois and Central Illinois is going to be major, given that infestation in some of those areas is just getting started. But anyway, to go back to the actual question that was asked, I can provide my contact information, and if folks are interested, they can get in touch with me and we can discuss the characteristics of the piece of land that they've got, and whether or not it would be a good fit for the program, and see if we can work something out. We're always interested in folks being proactive about this kind of thing, and we are now able to work with private landowners. That was not the case initially when the program began, but since they've changed that, it's really opened things up quite a bit. We have a lot more opportunity now.
All right, well, thank you, Nick. We're going to move on to our next speaker, and we do have a number of other questions that we would like to, if we have time at the end, we'll come back to you, Nick, with those questions, if it's all right.
Yeah, I'd be happy to.
Thank you. That was a great presentation. Let me move on to Dr. Fredric Miller. He is our next presenter. And while we're doing that, I think we can get his slides loaded. Dr. Miller is a professor of horticulture in the Department of Agriculture and Horticulture Sciences at Joliet Junior College in Joliet, Illinois. He's a senior scientist in entomology at the Morton Arboretum in Lisle, Illinois as well. He received his bachelor's in forest management from the University of Missouri and his master's in forest entomology from the University of Arkansas, and his PhD in urban forestry entomology with a minor in plant pathology from Iowa State University. Dr. Miller currently serves as the IDNR Forest Health Specialist and is a board-certified master arborist. And his research efforts at the Arboretum primarily focus on host plant resistance of ornamental woody plants for major economic insect pests, biological control of the emerald ash borer, and ash regeneration, and EAB population dynamics. He's actively involved in the International Society of Arboriculture and the Illinois chapter, which is the Illinois Arborists Association. So Fredric, we're going to turn the time over to you.
Thank you, Lydia. I trust everyone can hear me okay.
Yes, and Fredric, your slide is being shown, it's not being shown in display.
Oh, there we go. Sorry about that. There we go.
There you go. Perfect. Thank you.
Thank you. Welcome, everyone. Good to be with you all this afternoon. I wish we could be in person, but obviously we're having to make a lot of adjustments. Just want to say Nick has given you an excellent presentation on the biological control aspects of this. And I want to focus a little bit on some of the chemical management, but with the understanding that we will make a few general comments on some of the work we've done up here in Northern Illinois with the parasitoids, and the two seem to be working quite well together. And I noticed there were a couple of questions in there about whether the parasitoids are impacted by pesticides and so forth, but I'll get to that in just a minute. And then I want to talk a little bit about some work we did in the past using a fungus, Bavaria bassiana fungus, to also help control adults. Unfortunately, like Nick indicated, the virus really put the kibosh on things a lot this summer in terms of field work, but we have been able to do a little bit of local work up here in DuPage County, and I'll share that with you. And if we have time, I'm just going to squeeze this in, and Chris will probably get after me about this, but I just want to reiterate the fact that we need to be on the lookout for Asian longhorn beetle and also for the spotted lantern fly, but that'll be at the very end if we have time. So Nick's already shown you this map, and like he has indicated, I'm going to focus for all of you that are in the southern part of the state, central southern part of the state, as it relates to EAB, because that's where really a lot of the current activity is in terms of infestations and so forth. So like I said, I want to talk a little bit about chemical treatments, or what I like to call integrated chemical management. And I want to emphasize, like Nick has, there's not going to be one silver bullet here that's going to solve all of our problems for hardly any invasive and definitely EAB. So we really have to think about some chemical management up front to get things under control. Obviously bring in our parasitoids, our fungi, and then of course way down the road, and by that I mean probably a number of years will be before we have any resistant host plant resistance work, because it takes a long time to develop that. So I want to share with you kind of what I call a case study that was conducted up here in Northern Illinois. And I was involved in part of that process, and also it coordinates or is concurrent with the biological control project we're doing in DuPage County. But talk about how the pest biology, the economics of all of this, and community leadership really played a very important role in getting an integrated program going. So this is just a graph showing the representation of ash and other species of trees in this particular community. And you can notice, of course, maple and ash are at the top there in terms of numbers. And we were talking at the beginning, looking at around 17,000 ash trees just in the parkway public trees in this particular community. As many of you probably have heard, if you're doing estimates on ash, you can usually figure 2 to 3 times your parkway or public tree population for private trees, because it's a very popular tree species planted, of course, right after Dutch elm disease and replacing a lot of the American elms. And then if you look at most communities, the vast majority of this ash are green ash. White ash is about a quarter of that, and then very few European and other ash, which would be primarily blue. We don't really have much black ash here in Illinois, a lot of that up in Michigan, but most cases it's going to be green ash. So just to give you a timeline, EAB was first confirmed in this particular community in 2008. Now it was found in the state in 2006, out west of Chicago near St. Charles, out in Kane County. And then for a couple of years, I know the community attempted to do some citywide treatments, but it became very obvious very quickly that the insect had spread way beyond where we thought it was, and that's not unusual. That's been a very common theme all the way through. So that said, for those of you in central and southern Illinois, whether you're have private woodlands, forested areas, or you're managing parks, parkway trees, community trees, this thing can sit there for 6-8 years under the radar and you don't even know it's in the area. And that's what makes it so tough. So you really have to be vigilant in looking for it. And as Chris Evans's map that was shown a few minutes ago and that Nick showed, we still have areas where we have not officially confirmed it, but it's probably there in some fashion. Well, 2011 kind of became the tipping point. It became obvious that something was going to have to be done. And in this particular community, about a third of their third of their urban tree population were ash. So every third tree was an ash tree. So they basically looked at 2 options. One was to remove and replace all the trees over a period of 6 years, or to go in and what we call cull the herd, meaning basically you go in and you evaluate each ash tree to determine whether it's worth keeping or not. If it's not, you remove it or let it go and focus on treating the rest. The key here was not to try to save every ash tree. They wanted to save the trees that had the best possibility of surviving and could have future utility for the community. So again, I want to emphasize here, we're not talking about trying to save every ash tree in the community. So as a result of that, they set up a program where they did an annual assessment of each ash tree. Now, again, these are all parkway public trees. We're not talking about private. That's a whole nother can of worms, so to speak. They began treating the healthy ones, the ones that they felt were worth saving. They culled the herd, removed any that were in poor condition, they're not worth saving for whatever reason. Growth, structural integrity, trees that were leaning out over the street, those that had poor branch structure, all those sorts of things were eventually removed. And then, of course, coming back in and replacing with a diversity of tree species. So in 2012, the treatments began in earnest. This is where the public leadership got involved, community leadership. They developed EAB Awareness Week. They did a very good job of educating the citizens of the community to let them know what they were doing and the pros and cons of either doing nothing at all or proceeding with their treatment program. Tree condition survey was completed, and that's done every year. And approximately 16,000 parkways are treated. So you can see right off the bat, 1,000 trees didn't make the cut. Since that time, of course, treatments have continued, and they continue to do assessments and removing trees as is needed. This is basically what they used as their rating system. You can come up with whatever system you want. This is based on just general, good, fair, marginal, and poor. Dave Smitley at Michigan State has come up with a more formal evaluation system where he looks at percent canopy dieback. That's fine too, however specific you want to be. But the trees that they really focus on are primarily your good and fairs. If they get into the marginal or poor range, then there's the potential of cranking up the chainsaw and removing those trees. They basically went with three treatment products early on. And also I want to make clear here, I'm not endorsing any particular product. I'm just giving you the information on the trade names for these and of course the active ingredients. So there's a lot of different versions of the imidacloprid, which here is the Zytec. There's plenty of homeowner products as well as commercial products out there that are available. Safari is the valent product that is dinotefuran that can also be purchased under a green light label, which is a retail label. And then Triage, of course, which is out there, is the emamectin benzoate, which is sold as Triage, and there's several other trade names for that particular active ingredient. However, that one has to be used by a commercial arborist who's been trained, because it does take specific equipment for that. The Safari and the Zytec or the dinotefuran imidacloprid treatments, of course, can be done by homeowners as well. So how have the treatments held up? Well, for the imidacloprid, of course, it's giving good control, 71% tree survival. And again, that is an annual treatment. It has to be done every year. Emamectin benzoate is a every third year product. In other words, you can treat and then go for 2 years before you have to retreat. It's providing excellent control, 90% tree survival. The dinotefuran was discontinued after a few years because of the cost. They found it was more economical to use the imidacloprid active and the emamectin benzoate. Now, one of the things that you want to keep in mind, if you have large trees, and by that I mean anything over 15 inches in DBH, it's important that you consider treating those trees with Triage or emamectin benzoate. One of the things we learned early on the hard way was that we thought that the amount of product, active ingredient that you had to get into a tree was a linear relationship with tree size or DBH. And we found out very quickly that was not the case. It's actually exponential. So once you get over a 15-inch diameter tree, you have to use a much higher rate to achieve effective control. And that's particularly important with the imidacloprid. So you want to keep that in mind. If trees are in under that 15-inch DBH range, you can usually go with the imidacloprid and do fine, or even the dinotefuran if you want to go that route. But once they get into the upper teens, then you need to consider the emamectin benzoate product. What's going on now after about 10 years of treatment is that we're seeing that we're actually looking to move some of the triage trees to a 3-year treatment cycle. In other words, we're adding another year. We're playing around with this to see how these trees respond. And we're also doing the same thing with some of the imidacloprid treatments where we're skipping a year. I mentioned those treatments have to be applied on an annual basis in most cases. We're actually seeing now, because the EAB population has basically crashed in the area based on data that we've collected, that we're looking to pull some of these trees off treatment and only treating every other year. So my point here is this, I want to emphasize this. Once you start a treatment regimen, you are not locked in. You can play around with that, you can adjust it, you can do all sorts of things. So don't feel like that once you start treating trees with imidacloprid or emamectin benzoate that you're locked in. You're not. You can adjust that. You can see how your trees respond. And the other thing that you really want to keep in mind here is what's going on around your community. Now, I know a lot of you in southern and central Illinois are kind of isolated in the sense that you're not jammed up against another community like we are up here in the Chicago area. So that has a big advantage for you. One of the problems we had up here in Chicago was that, well, maybe one community was treating trees and the community next door was not. And so that was allowing a lot of beetles to come into the treated area and put additional pressure on those trees. So with you all in smaller communities, isolated communities, you're going to have a little more advantage there in that you can control what's going on around you. So keep that in mind as well. So how's it working? Well, we've got 80% ash tree survival. Again, as I said, the goal is not to save every tree, and there's been some that have not made it for whatever reason. It's just like any kind of a treatment regimen. Some patients make it, some don't, unfortunately. We're getting 94% survival of trees in a good and fair condition, and only about 4% tree loss per year. Marginal trees are showing significant damage. Those are ones that aren't responding very well. And of course, the poor trees are being removed in most cases. So this just gives you an idea, and I think this speaks volumes here in terms of the trees that, number of trees that have to be removed. And you'll notice they're, big, almost a doubling between 2011 and 12. It stayed high clear on through to 14, and then we've seen a very dramatic drop off. So we kind of have almost like a bell-shaped curve there. With the peak being in there between 2012 and 2014. The point I want to make here is if you sit here and you say, well, I'm not going to do anything, I'm just going to let nature run its course. Nature will run its course and EAB will dictate your schedule. But you're going to see you're going to get hammered very heavily in a very short period of time. So you look there between in about a 4 or 5 year period, you're getting hit with a lot of tree removals. And when you've got thousands of trees out there, or even hundreds of trees in some areas, you're going to be overwhelmed. And so, as we told people early on, you can either do it on EAB's schedule or you can do it on your schedule. And I would highly recommend the latter. So what happens is you get this compressed mortality issue where you go for a number of years, like you see in this graph, and this was developed by Dan Herms at Ohio State when he was there. You go for a number of years where it lays under the radar, you really don't see it, very low tree mortality, and then all of a sudden it takes off. And you can see in this graph from about, well, by 2006, you're up at 50% mortality and you're at 100% within just 3 or 4 years. So once the insect really begins to get rolling, it goes up very, very quickly, and you have very little time to respond. And of course, it takes a tremendous toll on your budgets. Now you can see here with this program that they were looking at, they estimated that treatment costs were much lower than letting the trees just die and removing them because of all the environmental and economic benefits that you get from trees. And Cliff Sadow's work at Purdue University with his tree calculator bears this out. So they estimated with these number of trees that there's approximately 2.7 million in tree benefits. And about $900,000 would be involved in treatments, removal, and replacement. So about a third of what it would cost in terms of benefits. Now you say, well, those are huge numbers. I agree, that's a lot of money. But you can take that and you can prorate that, or you can proportion that to your community. The numbers probably be lower, but proportionally, I think you'll see the treating and so forth is a much more economical way to go. Number of ways you can do this. The iTree assessment calculator can help you get some of these numbers if you need them when you go to talk to your decision makers in your community. Or if you're even deciding to possibly do this on your own private property, if you have some high value trees that you want to keep in your landscape. Approximately $30 per tree to treat one tree, at least in our area up here, and I know it'll be different down where some of you are down farther downstate. But you can anywhere from $700 to $800 to remove and replace that tree, not counting all of the benefits that you lose as having that tree gone, particularly if that tree's been there for 20, 30 years. So just some comparisons here. The removal replacement plan, you'd have to take out all the ash trees. We'd be looking at 2,800 trees per year for 6 years to get those numbers down. Well, you look back on that table I just showed you, and we never got close to 2,800 trees. We were in about a fourth of that with the program that they had, but imagine fourfold and doing that for 6 years in a row at about 1.7 million. They also compared that with what it would cost if they did a treatment plan. You can see it ran about 850,000 per year. Again, no argument, lots of money there, but 850,000 is a lot better than 1.7 million. and the fact that the regimen could be maintained, they estimated out until about 2028. So they were looking at about almost a 20-year period of time there where they could afford to treat trees and still balance out on the removal replacement costs. So if you look at this graphically, you can see here that when they first started, EB was first discovered, that within just a few years, they were going to be at 100% mortality of all their ash trees, as opposed to the treatment program where you came in at about 40% of the cost. And of course, you're looking at around 5 to 6 million there, excuse me, 4 to 5 million versus up to 12. But spread out over a much longer period. Yes, that's a lot of money, but spread out over many, many years compared to getting that all in just a short period of time. So as I mentioned, they are currently, every year we reassess things. We look at maps. They have, of course, everything very well mapped within the community, so we know exactly where all the treated trees are. We're beginning to move about 25% of those off treatment. And we're going to a 3-year schedule, and they estimate right there they're going to save 20% in treatment costs. We also are looking at other, like the imidacloprid, like I indicated, where we're actually skipping a year on those and monitoring those. I can tell you, we started this 2 years ago, taking trees off treatment, and they are all looking very good at this point, because I hope with some of those assessments and looking at those, they're holding up quite well. There's still some that aren't responding and they'll be removed, but the vast majority of them doing quite well. They estimate if they quit treating, we could get right back into a very difficult situation. And of course, all that investment that you had would be lost very quickly. So what are they doing now? They're definitely looking at hotspots, and we've got several in the community where they join other communities where treatment was not done or not done very effectively. And so they're keeping an eye on those areas and you know, watching those much more closely. They're replacing and removing where needed. Of course, all of this is leading to a much more diverse urban forest. We didn't seem to learn our lesson from the Dutch elm disease days back in the 60s and 70s. And so hopefully we'll learn more from this now and not be going back and planting monocultures or allowing folks to come in and just plant one species of tree. And of course, as I indicated, we're going to continue to take each year probably more and more trees off treatment, and we're going to monitor them. If some of them don't respond real well, then we may put them back on treatment. But hopefully, with most of the trees in an area treated, what Deb McCullough at Michigan State calls the herd mentality, or the herd, you've heard about this with the virus. If you don't necessarily have to treat the whole population, but if you treat a good number of the population, that can protect everybody. So we're talking about the same concept here with emerald ash borer, just like we are with COVID. So that's where we are with the chemical treatments. Again, I would encourage you to think about your inventory, look at what you have, think about how many trees you want to conserve. And even if you eventually wind up taking them down, you have spread out that treatment removal cycle that is going to make it much more manageable and much easier to plan budget-wise, financially, as opposed to just getting hit hard all at one time. And of course, along the way, you still have the flexibility to do, your routine pruning, tree removals, anything else you need to do for your tree population. If you talk to people that have been overwhelmed with this, they'll tell you all it is, you live and die and breathe EAB for years, because that's all you can manage. You have no time to do anything else. So if nothing else, for your own sanity, it's worth it. Nick talked very extensively about the biocontrol. I'm just going to make a few general statements here, because that really wasn't the thrust of my talk. We have been doing parasitoid releases up here in a forest preserve in the DuPage County, Will County area. It's proving to be very effective. We started that in 2015, and we're seeing, as Nick indicated, the TETS is established very quickly within the first season, and actually dispersed up to over a mile from where we released them. So it's done very well. The polar vortex took a little toll on them, kind of knocked them down a little bit. But they've bounced back, and we're seeing good numbers this year. We'd like to think that the combination of our aggressive tree treatments along with the parasitoids has really helped bring the EAB population down to basically levels that we feel that the population has crashed. Now, the big question now is, will they rebound? We'll have to wait and see. But as Nick has indicated, we're hoping that the parasitoids and other things that we're doing will help keep that EAB population low enough where we won't see the really large scale tree death like we have in the past. If you think about it, EAB was out there for 6 to 8 years. The green, the white ash were fighting that insect and trying to survive, and did that for a number of years. But once the beetle populations, of course, exploded on us, then they were not able to fight it off. So if we can keep those beetle populations low enough, I think our trees can tolerate that and continue to do okay. One other biological control project that we've been involved with is involved using a fungus. We've been working with a company out of Canada that has been using this up there. They're much more restrictive on what chemical treatments can be used up there. So they've had to look at other options for managing their ash populations. So in 2017, we started a couple of studies, one in Quincy and one in Carbondale. We've had some glitches along the way. Of course, the big glitch was this summer. We couldn't do anything because they couldn't come down from Canada and we weren't able to really do a lot of field work. So we kind of lost 2020, but we did get some work done in 2018 and 2019, and does look promising using this fungus. So again, we're talking about a multiple tactics here. If you're just getting started with EAB, keep them under control, do your chemical management. Then if you can work into some biological control, then you are going to be in much better shape. And as Nick indicated, with any biological control agent, you can't expect it to come in and do an effective job if your pest population is out of control. I don't care whether it's in a greenhouse or in anywhere else. You have to keep the pest population low enough so your biological control agents have a chance to get in and be effective. And that's really going to be the key for you all in central and southern Illinois. If you can get in there early enough and keep your EAB populations low enough, you can bring in your biological control agents and they can pick up the slack and hopefully take over. So basically what we have is a fungus here that we use this green funnel trap over here on the left-hand side. Has what we call, at the very bottom, you see this kind of 2 inverted cones down here. This is what we call a contamination chamber. They basically put the fungus in there. The beetles are drawn into the trap just like they normally are when we have, and we do have a pheromone on there. So this is what we've used in the past for monitoring for EAB. So we're using the same style trap. They come in, find their way down through those funnels into the contamination chamber and then leave. They leave the trap alive, but in the process, they pick up the spores of the fungus. And then of course, during the mating process, they share these spores with their significant other, and hopefully that becomes a lethal situation. So on the right here, you can see evidence here of the fungus actually colonizing the adult EAB. So these are suspended in trees, and it's a dissemination device. We also put up regular panel traps like we used to use, what we call Debarking traps, the big purple traps, to monitor for how many beetles we're actually catching in the area, along with the ones that are contaminated. And the intent of this is to contaminate or infect the beetles when they're feeding. They have to go through about a 10 to 14 day feeding maturation period before they start mating. And so in the process, they mingle and get acquainted and all that sort of thing. And that's when they pass the fungus to each other. But it is this Bavaria bassiana. It's been around. It's a naturally occurring fungus. It's been around for years, used in greenhouses for a lot of your greenhouse pests. And now we're beginning to develop it for some of our forest pests. So here on the left here, you have the pads, these fungal pads or pouches, as they call them, that go into the chamber. And then, of course, you have an infected EAB over here on the right. Here's the setup. You have your the left-hand picture there. You've got your typical 12-unit funnel trap, lingering funnel with a chamber at the bottom. And then here's your green panel trap that's being used to determine population levels. And we've been getting pretty high infection rates with this. And I'm hoping that in 21 we can get back on track and be able to do some more field work. So in looking at some of the work out of Canada, if you look at the number of eggs laid here on the vertical axis, under natural conditions, of course, you can see the pipe A here, the line just takes off and goes out through the roof. The 2017 protocol or work that they did is line B here, the blue one. You can see it kept it much lower. And then, of course, the objective is the C line down here, the red line, to get it as low as possible. But it is very encouraging. And again, it's not going to be the only thing that we use. Obviously, we're going to have the parasitoids out there. We'll probably have to throw in a little bit of chemical management with that, but that's the whole point of an integrated program. You don't rely on just one method. So they're continuing to work on registration with EPA here in the States. We want to try to continue with some of our protocols, see if we can get more data and get a little bit finer handle on how many traps you would need in an area and so forth to get the desired infection rates. Quickly, I think I'm okay so far. If not, let me know, Lydia, if I'm running over here. We've been doing some work at the Morton Arboretum with host plant resistance. Of course, they're doing work. The U.S. Forest Service is doing work as well on this. But we have a nice luxury there at the Arboretum where we have a very extensive ash collection. And a lot of these were brought over from China, from Asia early on. And so we're having a chance to do a little bit of work with that. So I just want to hit some highlights. One of the things that you look at, of course, with any wood-boring beetle, excuse me, is the amount of phloem that they consume, because that's what they live on. And you can see here, of course, Pennsylvanica is your green ash. And then we've got Manchurica, which is an Asian species, which was brought over early to hybridize with the black ash. But that didn't pan out too well because they seemed to like it as well as they did the black ash by itself. Chinensis, Stylos, and Chinensis ranchophylla are all three Asian species. And you can see that they definitely consumed less of the phloem, these would be the larvae now, EAB larvae, on Chinensis, Stylosa, and Chinensis ranchophylla. So those look fairly promising as potential in terms of larval feeding and larval damage. Then if you look at survival, same kind of pattern, much lower survival on those three. We're talking below 20% in most cases, and that's up to 60 days. So some of the early work that was done on things like black walnut and wingnut, and some of those, the larvae would pour in, they'd go a short distance, and then they would die, and there would not be any more gallery activity. So kind of the same thing here. Some of them do survive, but most of them don't, as compared to green ash, where you've got over almost 60% survival rates. So that was encouraging as well. And with any insect pest problem, as long as you break the life cycle, stop it from reproducing, that's really all you need to do. We also looked at leaf tissue feeding. This is important because of the fact that, like I said, the adults have to feed for a period of about 2 weeks. So if the adults don't feed long enough, sufficiently enough, then that's also going to impact their ability to reproduce. So the solid bars to the left there are all Asian species. We had 2 or 3 European species with the dashed lines, and then the hollow bars there would be, of course, our North American species that we used as a comparison. But you can see, again, quite a bit of variety, but the 2 down here, a couple of the Asian species as well, look pretty promising for adult feeding. But again, just because they feed a lot does not necessarily they're going to reproduce or survive. And we looked at the amount of frass. Of course, frass is a combination of insect poop and plant material. And you can see, again, the lower, less fed upon species produce much less frass, so they're not utilizing that food very well compared to some of the others. So we do have several Asian species in here that look promising from a plant breeding standpoint. Of course, you also have to look at their ornamental qualities and their growth habit, a variety of different other factors before they actually are going to wind up in the trade. But at least we do appear to have a few that have some potential. So just to wrap up here a little bit, we're seeing where the chemical treatment is quite beneficial, it's effective. That wasn't the case early on. We did have a lot of inconsistencies early on with chemical treatments, but we've figured that out. So we continue to monitor by taking branch samples in the springtime. We're looking at hot spots, and of course, paying attention to what other people do in other communities. As I mentioned earlier, yeah, get biological control in there as early as you can. We have seen some of the same native species that Nick referred to, but again, they're contributing a fairly small amount because they are generalists. They're not just homing in on EAB, but they are part of the mix. And as I mentioned, we do have some promising ash species down the road. I'm just going to hit this real quick. We had several calls in here from nursery growers and other folks about some of the ambrosias. One of the things we're looking at through our forest health monitoring is laurel wilt. That is a fungal pathogen that is attacking a lot of the laurel, red bay laurel and things in the southeast, but it also hits sassafras. So those of you in central southern Illinois, this is another potential host for this fungus. We're looking at ambrosia beetles and also the bark beetles that are transmitting TCD. Just a couple of comparisons here. Ambrosia beetles are primarily fungal farmers. They do not actually consume the cambium of the trees, but they grow the fungus in their galleries to feed their young, and they're very reliant on that. So they're obligate fungal farmers, as compared to bark beetles, where they feed primarily on the cambial regions of the host and do a lot more damage. So both of these working together can be pretty destructive of some of our woody plants. And of course, the big problem with the laurel wilt, it's transmitted or vectored by this red bay ambrosia beetle, which we've been trapping for this last summer and will continue next summer, particularly in southern Illinois. So we put out pheromone traps for that and see if we can find any. But this is a very aggressive fungus that is killing a lot of the Lauraceae family members like red bay and sassafras and so forth. But the plant actually overreacts. It's kind of a similar thing to what we saw with Dutch elm disease. The plant tries to isolate the fungus by producing tyloses, and usually the fungus moves faster in the plant than the plant can keep up with it. And so it kind of, in some respects, kind of the plant's its own worst enemy in that respect. But the fungus is carried by this red bay ambrosia beetle. It is native to Southeast Asia, so it is an exotic invasive. And you can see here it's killed over 500,000 red bay. And that's a very important species for the southern forest ecosystems. We also have them coming in on stress plants. This up in the upper right here is Germanus, and then Saxoni down here at the bottom. These are all beetles that come in on stress plants, flooding, drought. This is where we're seeing some of this in tree nurseries. They also will attack fruit trees. So this is something you want to keep an eye out for. We do trap for these here in Illinois on a regular basis. And we do tend to see more Germanus up north. We see more of the Saxoni and the Cresculus down south.
Hey, Fredric, we're running a little late.
Okay.
So I think we're
gonna wrap it right there then.
Yeah, that makes sense.
Thanks.
Well, yeah, great. Thank you so much for the presentation.
Certainly.
If you're okay, so what I'll do is just wrap up now, and if people want to stay around and we've got a few questions come in, then we'll ask questions. So
That'll be fine.
Okay. So I just want to thank everybody for attending. Again, if you want to stick around, we'll answer some questions. Feel free to ask questions in the chat box. But for those of you that are IFA members, we hope to see you tomorrow at our annual business meeting. Look for an email from Zach coming up shortly on that. But Fredric, we did have a couple questions that came in already.
Okay.
And one of them was basically, does ash tree density, so the number of ash trees per acre, or how close they are together, does that predict or influence kind of the likelihood that EAB will get there, or the severity? And is there any way to kind of mitigate that with planting other trees or spreading ash out?
Well, yeah, definitely ash tree density plays a role. We saw this very extensively in the urban areas up here, and I think that's probably why EAB just went crazy, because it looked out over the state of Illinois and said so many ash trees in so little time. But yeah, the more hosts you have, then that provides plenty of resource there for the insect to, really explode and reproduce. So We had some communities up here in Northeast Illinois that maybe only had 10 to 15% of their tree population were ash. They didn't suffer nearly as much as communities that had 30 and 40%. So for some of you down in Southern Illinois, Central Illinois, you may have a few ash trees here and there. If you want to save those and they're worth saving, that would be a good approach early on, but you're probably not going to see as much devastation as we saw up here because we just had such high populations. Now, I am, like Nick said, I'm seeing a lot of regeneration coming back in. It's been amazing in the last few years, both seedlings and sprouting along the interstates, a lot of your forest preserves, forest edges. And so you want to keep an eye on that because that's the next phase here. We don't know how much the insect will build back up with all this regeneration. That's the next phase of this thing. But yeah, your ash population will have a lot to do with what your numbers are.
Okay, great. Earlier on, you said something about how emerald ash borer may sit dormant for 6 years and unseen for 6 or more years in an area. And somebody asked basically to explain that, like exactly what's happening. Why are they dormant or not seen,
Right, dormant may not be the best word there. They're definitely active. Well, it gets back to what I said earlier about the trees being able to really fight off the insect. Yes, green ash, white ash, blue, black are all susceptible. All the North American species are susceptible to EAB. Green ash and blue and black are highly susceptible. We see white ash appears to have a little bit of resistance, and blue ash even more. But they all, if they're hit hard enough, they're eventually going to succumb. So what I meant by that is, as with the low EAB populations, you may not see a lot of visual signs of dieback, decline. That's what happened in Michigan. They saw ash trees dying, but they didn't know what was causing it, and all of a sudden they discovered it was this, beetle. So that's what makes it tough with EAB, because it can be there under the radar and you don't know it's there, and then all of a sudden it just takes off on you. Sure. And so that's why monitoring, checking. Wintertime's a good time to check. Nick mentioned the woodpeckers. You go out there and you look for woodpecker activity, bark splitting. Those kinds of things are good indicators in the winter to tell you, okay, I may have EAB here. Of course, you want to get it confirmed, and then you need to start thinking about monitoring for that. But that's kind of what I was saying. It takes a while for it to build up, but when it does, and you saw where it changed there in about 2011, 2012 for us up here, then it just exploded.
All righty. We have one last question. If somebody sees patches of sassafras in Illinois dying, and they're concerned about this laurel wilt, what should they do? What do you recommend? What's the steps for them to take to kind of alert people or do something about it?
Well, the first thing would be very helpful. If you want to contact me directly, that would be fine, because I'll definitely pass that word on to Scott Shermer or whoever with the Department of Ag, or you can go right to the Department of Ag, but definitely contact someone, Extension, folks, your local district forester, myself, anyone, because we definitely want to investigate that. Again, sassafras is not going to be like a major hardwood species in terms of value, but it does play an important role in the southern ecosystem, forest ecosystem, and I like sassafras teas, so no other reason there. But yeah, please contact an official of some sort so we can get a handle on that and document that, because that'll be very important. We have to have the wilted plants, the symptomatic plants, just like with TCD and walnut twig beetle, same thing. We need to have that documentation to actually show that it's there. One final comment. I know one person asked about the compatibility of the insecticides and the parasitoids. We have not seen any problems. We have found actually reared parasitoids out of treated trees and untreated trees. So there doesn't appear to be any issues there with the parasitoids attacking EAB larvae that are in treated trees. So that's a good thing, because otherwise that kind of cancels everything out. But just wanted to throw that in there.
Sure, great. That's good information. All right, Fredric and Nick, both, I want to thank you both for all the presentations and the questions answered today. It was really great presentations.
Thank you.
Great. And I want to thank just everybody again for attending our Illinois Forestry Association virtual annual conference this year.